Well, good afternoon, everybody. It's lovely for you to join us for the final session. Following genetics and environment, I think it's appropriate that we conclude with aging, which is the culmination of these factors. We've got a great panel. I have to say I'm looking forward to their talks very much. We will start with Paul Matthews from Imperial College. Now, Paul has more letters after his name than my short-term memory can cope with, but he's one of the UK's most influential biomedical scientists and is in an excellent position to give us an introductory talk on how UK Biobank can transform aging research. Over to you, Paul.
Thank you, John. As always, it's an overgenerous introduction, but thank you. It's a pleasure to be here this afternoon to speak about enhancing UK Biobank for dementia research. The agenda of dementia is an urgent one. It is a major global threat. Dementia currently affects more than 850,000 people in the UK, and by 2040, that's going to rise to well over just getting the clicker to work. There we are. With one diagnosis every 3 minutes, this should rise to well over 1.6 million by 2040. This is as I said, an urgent global problem. More than 55 million people in the world live with dementia, and to make matters worse, 60% of these are found in lower and middle income countries. UK Biobank presents a special opportunity for helping us understand this disease. We're following 500,000 people with exquisite phenotyping, as you've heard through the day, as well as genetics. There's been 15 years of follow-up to date, and many more are expected, and the data is open to everyone around the world. What this means is we are able to watch dementia evolve from its earliest stages in this population in really unique sizes. The richness of the associated data is allowing us to understand in very precise detail the associations of lifestyle and environmental factors. For example, in this paper recently published, there was a careful analysis of the role of a range of comorbidities and lifestyle factors on risks of dementia. Here you see one of the most important of these, which is the association with hypertension. See on the far left, the association of hazard risk for dementia with systolic blood pressure, showing the increased rates of the disease at low and high blood pressures, but more importantly, with the scale of the data that's becoming available to us in UK Biobank, we can perform Mendelian randomization studies to begin to explore the causal associations, really for the first time providing very strong evidence that blood pressure is causally related to high BP, to high rates of dementia. This gives us some hope that efforts to control dementia by reducing blood pressure may be effective in the future. The large numbers are also allowing us to explore the genetic basis of Alzheimer's disease more fully, just as you've just heard so elegantly for the heart and thoracic aortic aneurysms. There have been several studies of Alzheimer's disease, but what UK Biobank has allowed for the first time is a large-scale study of Alzheimer's by heritability, which came up not only with the genes that have been associated in the past, but also extended understanding of the disease with identification of new genes, notably affecting the angiotensin converting enzyme that we've heard so much about over the COVID period, opening up a very intriguing line of inquiry regarding the relationships between COVID risk and brain risk in the disease. Now, the scale of the data that people are developing within UK Biobank in conjunction with that already available is allowing for the first time a dissection of risk, genetic risk contributions to the disease by pathway, showing, for example, that activation of immune response pathways in the brain via microglial and astrocyte responsivity is particularly likely to be important in the late onset sporadic disease. It also is highlighting increasing importance of lipid pathways associated with genetic risk as the disease progresses. Now, over time, this resource is only going to become more informative. Within the 500,000 people being studied, while the number of incident cases thus far is relatively modest, only some thousands, by 2030, there should be almost 40,000 people within the population who will have developed Alzheimer's disease. The people we are now observing are allowing us to observe the disease in its cellular phase as it evolves. One of the new tools that has been introduced over the last few years to help us understand the disease more fully and its comorbidities and their relationships has been the imaging enhancement that was introduced in 2017. Since its initial introduction, more than 50,000 people have already been scanned, brain, cardiac, abdominal scans using MRI, carotid ultrasound, full body DEXA, and a whole range of baseline assessments, which you've heard reference to through the day. This is providing an extraordinary range of tools to begin to explore causal models for prediction of endophenotypes in the imaging and other markers, and to relate those together to disease outcomes in ways that help us understand mechanistically how patients are stratified and how the disease evolves in different subgroups within the population at large. Let me highlight just a couple of the very special insights that are coming from the imaging enhancement. One is that we're able to look at those disease-associated phenotypes that really are only apparent through imaging, such as the marks of white matter hyperintensity in the brain that provide indices of small vessel disease, one of the major contributions, one of the two major contributions to dementia risk. Here's an association study, one of the most complete to date, that was done by colleagues in Bordeaux, highlighting the gene associations not only with white matter hyperintensities, but those but the relation of these to other diseases. You can't. I won't go into the details on the figure to the left, but refer you to the paper. However, if you look to the right, two-sample Mendelian randomization in association with other elements of this study was enabled by the genetic links, and it highlighted a variety of causal relationships with other diseases. One of the most important is at the bottom, emphasizing the relationship between white matter hyperintensity load and small vessel disease and Alzheimer's disease for the first time in a very clear way, as was suggested inferentially from the pathology. Moving on, looking at the we'll hear more about the body phenotype soon, but imaging of the body allows looking at a variety of factors, one of the most important of which is fat distribution, where you can see in these images of a variety of UK Biobank population, subcutaneous fat in blue, visceral fat in red, lean tissue in other bright colors. The combination of these can be used in predictive models to look at risks of coronary heart disease, type 2 diabetes, CHD, and T2D, as shown in the plot at the bottom. But what's really important for this talk is not only can one use these to understand systemic disease phenotypes, but it's become clear that the distribution of fat and lean mass, here reflected in BMI, is related to progressive brain volume loss with aging. Here is a map that was generated in our group looking at the rate at which brain volume loss is accelerated with high BMI. Here is a plot of the rate of change of gray matter thickness across the brain as a consequence of this and related comorbid disease factors. And here you see in a more recent publication, a bidirectional Mendelian randomization study associating causally metabolically unfavorable high body fat with increased risk of dementia and brain volume loss with aging. The availability of imaging data directly from UK Biobank has allowed innovation by the community. Here's a beautiful study from Carla Miller's group showing how quantitative magnetic susceptibility maps, which provide indices of iron deposition in the brain, have been generated as a secondary output from the UK Biobank data used in a genetic association study, which has highlighted a number of genes, amongst which is an important and interesting gene illustrated here for its brain-wide associations, the gene associated with hemochromatosis. This is of special interest because the hemochromatosis gene has been linked to the risk allele has been linked to a decrease in HMG-CoA and progressive brain loss with time and dementia. We have used typically brain atrophy around the hippocampus as an index of progression of cognitive impairment and dementia in the clinic. What UK Biobank and other large data sets is providing is ways of using much more detail in conjunction with AI methods in order to inform on risk. Here's a beautiful example from Salad's lab of the way in which this has been used to explore the increasing risk prediction to over 90% predictive capacity. I'm going to have to start closing, but I want to highlight these same associations and the advances in image analysis methods that are enabled, as we just saw on the previous slide, are beginning to not only to define cardiac disease risk factors, but relating cardiac, in this case, aortic disease risk factors to those in the brain, showing via endothelial gene-inferred metabolic circuits. There are many things that we can do in the UK Biobank to enhance it going forward with for dementia, with greater cognitive testing, proteomics, both of which are coming towards the end of the year. Future work for epigenetics, increasing our phenotyping of behavior with digital tools and responses in the brain with portable brain wave or electrophysiological monitors. And of course, there are a whole range of new biological measures that can be performed on biofluids such as exosome analyses or those of the microbiome. I want to close by thanking many people, but most importantly, I don't want to forget the many, many people who are participating in UK Biobank and so generously providing this data to the whole community. Thank you, John.
Well, thank you very much. An excellent overview as ever. Now, we're very privileged to have Nick Van Bruggen from Calico. If I had to choose a word to characterize Nick's career, it would be innovation. He's had 25 years in drug development. He's introduced imaging, and I can't think of anybody better to describe the repeat imaging in UK Biobank. Nick, over to you.
Great, many thanks, John, and thank the organisers for inviting me. So I was asked, I work at Calico. I was asked to give a, how do you change the pin here? I was asked to give a quick overview of how Calico is thinking about using the deep and rich phenotyping which is available in UK Biobank, specifically the imaging. By a disclosure here, the repeat imaging is an initiative which we're just about to embark upon. This will be funded in collaboration by Calico, CZI and the MRC, and more of that later. So I thought by way of introduction, I'll just say who Calico is. So Calico is an Alphabet funded research and development organization. Our mission and our focus is understanding the aging process which underlies disease in human aging. Calico is, its intent is to develop therapeutics which can either treat the aging process or treat age-related diseases. So why study aging? I think this is probably self-evident. With the aging population, as outlined on the left, there's a real need to understand the processes which contribute to the aging process itself. This figure on the right here simply shows the CDC data, which is plotting the log mortality hazard, showing an exponential increase in the risk of disease for a number of the major diseases of the general population. So aging is the dominant risk factor for the world's most prevalent diseases. This idea of mortality hazard, of course, is not new. The decline of physiology in the general population, which contributes to the hazard, was described back in the 1800s by Gompertz. So the Gompertz law of mortality is the human death rate given by the factor B in the exponential curve is the sum of the age-dependent components and increases with age. So our challenge here is to understand what those components are, and one way to do that is to do deep phenotyping. This is a quick aside, but I thought it was interesting. A paper which we published a few years back now in eLife by Graham Rudy and Sherry Buffenstein shows that the Gompertz increase in mortality hazard is quite common across mammalian species. This shows it for mouse, for human, for horse, for example. It's worth noting that studies which they did at Calico shows that the long-ribbed rodent, in this case the naked mole rat, does not obey the laws of Gompertz increase in mortality. In fact, you can see here the curve is very flat, and so the death rate within the naked mole rat appears to be stochastic, and does not follow the Gompertz laws. So that gives you at least some hope that there are fundamental processes which contribute to the rate of aging which can be perturbed. So to put this into practice, we published a review article a few years now. It was a part of the big data review which was published in JCI. And to cut a long story short, if we want to look at the process rather than the disease state, we need precision phenotyping to measure that. And then we need to model on those precision measurements. So UK Biobank, because of its imaging cohorts, extended cohorts, and because it is a cross-sectional analysis with with outcome measures from an aging population is kind of ideal for us to do this. Okay, so here's an example. It's a fairly straightforward example. I believe it's been shown to the UK Biobank audience before, but I just wanted to use it as a way to illustrate how we think about getting a handle on the underlying processes which will contribute to aging in the human population. So a few years back, a couple of scientists at Calico, Eugene and Anurek, looked at a database which was mortality in its origin. It's called Mr. Oz, and they were measuring the increase of fractures in men as a function of age. And because of mortality, a database, it has a 16-year outcome, and you can look at the survival in that database and correlate that with reasons for having an increased mortality. So the aim of this study was simply to use planar X-ray, and that planar X-ray was designed to measure bone density to look at fracture rate. But of course, the X-ray technique itself is also sensitive to things like vessel calcification. And the example on the left there simply illustrates that. You can see the vertebra, but the box is highlighting a bone-like phenotype associated with the aorta. And that bone-like phenotype, when you run the analysis as survival curves, you find that that has a dramatic increase on mortality rates. So that's an interesting study and points us in the direction of an underlying pathophysiology which is contributing to aging. But of course, we really need orthogonal data to understand the biological process there, and the UK Biobank offers that. So within the UK Biobank, DEXA scans were acquired, and I want to make the point that the DEXA wasn't acquired in order to make this measurement. This is just the fact that we have very large N, and we can automate the analysis of very large N using deep learning techniques. We're able simply to pull out that measurement from the noise. So the figure on the left there shows that. It shows the spine with the vertebra, which is attenuated in X-ray signal, of course, but then you also see in that little box what looks like it's within the noise, but it's actually the calcification of the aorta. So we can apply these automated techniques to quantify the level of calcification from DEXA scanning. And when you do that, you can start to get a handle on the underlying contributions of this pathophysiology to the disease state, as well as the molecular mechanisms which contribute to that pathophysiology. Again, one of the things which I think is so elegant about this is that these DEXA scans were not designed to make this measurement, but again, the beauty of the UK Biobank combined with our computational analysis techniques that we can extract the signal from the noise means there's a huge number of parameters which we can pull out, an infinite number of parameters almost which we can pull out from these images. And this, of course, is a very straightforward one, but a very nice example. So the DEXA scanning identifies the degree of calcification, and then we can use the outcome measures which are available within UK Biobank and integrated modeling to look at how that contributes to diseases of aging, which is shown in the middle panel there. And then also we can do a comprehensive analysis of the genetics and the biomarkers in order to understand the major drivers. And in this particular case, it was quite consistent that this calcification was associated with transcription regulation and an osteogenic conversion within the vasculature. Okay, another example using the same principles where we can use precision imaging in order to extract meaningful and precise information about biological processes which underpin the aging process. This is a study which was recently published from Calico scientists in collaboration with our with collaborators at Westminster. We know, and we've previously shown, that iron accumulates with age. And the question really is how does that influence disease-related processes? So the UK Biobank has, and again, the study wasn't designed to make this measurement, but because we have susceptibility mapping, we can infer the degree of our iron within the organs. So this is one particular example. It shows the iron within the spleen. That enables us to take precision measurement and then do very precise genetic association with that. And again, the major genes we pull out are associated with loss of erythrocyte membrane proteins and integrity. And then again, we can correlate that with disease outcomes to understand how that specific pathophysiology contributes to the aging process. So those are specific examples, but those are cross-sectional studies within the UK Biobank. What we now want to do is to extend that analysis and do repeat measures. Of course, the advantage of doing repeat measures is, I think, fairly obvious. You get increased statistical power. You can allow for pre-existing risk pathologies, which often contaminate our measurements, to be to be frank. But I think most excitingly, you can do voxel and vertex-wise analysis. So we can now start to map those processes, which we think contribute to the aging process. So here we just have a quick example from a pilot study, again, performed in collaboration with the group at Westminster, and Jimmy Bell provided this slide. This is 3,000 people, and this is over a 2-year period. And this figure on the right there simply shows a number of different parameters which we can, out of many, which we can pull out from this. So very quickly, just to wrap up in the last remaining minute or so, just to illustrate how that longitudinal data can be used. So longitudinal data, this is very early in the process, and so we only have very early studies, but we have published a 2-year observation. And this is a simple example. Here we have the body mass index at baseline versus body mass index 2 years later when these people were re-imaged. And most of those data points lie on the unity line. That said, there are some which increase the variance of that measurement, and they increase the variance of the measurement because they have a change in body fat, which you then go on and measure. And so one of those is shown in figure D, and one of those is shown in figure B. So this is really reflecting a 2-year period, so it's not a very long period of time. You can identify the underlying changes in adiposity, which contributes to maybe that lifestyle or health style change within UK Biobank. That gives us real encouragement that within a fairly short period of time, you can measure fairly dramatic, precise imaging phenotypes associated with that. That's clearly a lifestyle change, but also we're looking for changes which are time-dependent and aging pathophysiology. So here's another example where the team at Westminster went in and measured the muscles associated with the compound muscles associated with the inner hip. These are a good measure. The volume of those muscles is a good measure of sarcopenia, for example. And here we see that the majority of participants in that over that time period, that 2-year time period, have a reduction in the total volume of those muscles, again reflecting probably a sarcopenia progression and therefore an age-related pathophysiology. So that reminds me just to thank the organisers and thank everybody who's put together the UK Biobank, and to thank the participants in UK Biobank who have made these studies possible.
So Nick, thank you very much indeed. You know, the power of precision imaging, I think, is yet to be fully realised, but this is certainly, you know, very encouraging and exciting. And it's fitting, therefore, that we go into further precision imaging with Pearse Keane from Moorfields Eye Hospital. Pearse is a clinical scientist with NIHR, and he initiated the collaboration between Moorfields and DeepMind, Google DeepMind. So Pearse, over to you.
Thank you, John. So I'm going to talk about an area that we've called oculomics. And this is using the eye as a window to the body, taking advantage of some of the latest advances in retinal imaging, plus the latest advances in artificial intelligence. And as John has said, I'm a consultant ophthalmologist at Moorfields Eye Hospital, where I work as a retina specialist. So I particularly specialize in conditions such as age-related macular degeneration. And I'm also a professor of artificial medical intelligence at University College London. And although I'm not a computer scientist, I'm not an engineer, I'm a clinician, I've somehow come to find myself leading a multidisciplinary research group that aims to develop and apply AI in healthcare using ophthalmology as an exemplar for the medical specialties. I'm also fortunate to be funded by UK Research and Innovation, and I should disclose that I have acted as a consultant for Google and for DeepMind. So when we talk about using the eye as a window to the rest of the body, of course, this is something that has been known that it's possible to do for more than 100 years. The exciting thing now, though, is that with the latest advances in artificial intelligence, in particular a technique referred to as deep learning, we can look at a retinal photograph, such as the one that we see here, and using deep learning, we say, this is a woman. And using deep learning, we can say she's 58 years old. And using deep learning, we can say whether she smokes or not, whether she's diabetic or not. And using deep learning, we can even say her BMI, her body mass index, is around 25. and her blood pressure is around 150/85. Now you may say, well, you know, what's the point in using artificial intelligence to tell if it's a man or a woman from a retinal photograph? There are easier ways to make such a determination. But of course, that misses the point of why this has got everybody within the ophthalmic community so excited about this. Because, you know, although I've been looking at retinal images obsessively for probably about 20 years now, I cannot begin to say whether it's a man or a woman just from looking at a retinal photograph. And I cannot begin to give a close estimate of the age of the person by looking at a retinal photograph. Now, UK Biobank has played a pivotal role in much of the recent advances and in this field that we're terming oculomics. And in fact, in UK Biobank, there's about 117,000 people in the eye and vision component of UK Biobank. And of these, about 68,000 patients had retinal photographs and OCT imaging done, optical coherence tomography imaging. And in fact, it was a team from Google Brain publishing in Nature Biomedical Engineering in 2018 who were the first people to really describe applying deep learning to predict things like biological sex and age, as well as a range of cardiovascular risk factors from the UK Biobank data. There's been a number of other publications that have built on this work since that time. Now, why is the eye, and in particular the retina, a perfect window to systemic health, or a very good window at least? Well, we can, within the retina, we can directly visualize the microvasculature of the retina. And of course, famously, the retina is an approachable part of the brain. So it's the only part of the central nervous system that we can directly visualize without opening up someone's head. And so with advances in imaging such as optical coherence tomography, we can actually visualize the layers of the retina and the walls of the eye with micrometer level resolution. So potentially order of magnitude better resolution than an MRI or a CT scanner. Now, as a result of these advances, my colleague Alistair Denniston coined the term oculomics. And as I've already alluded to, this is when we combine advanced retinal imaging with big data and artificial intelligence. And we wrote about this in the journal TVST in 2020, if anyone is interested in learning a little bit more. Now, in terms of the initial work that we've done with UK Biobank, much of this has been led by the UK Biobank Eye and Vision Consortium, led by Professor Paul Foster, and much of the OCT work has been led by my colleague at Moorfields, Praveen Patel. And so a lot of the initial work that they've been doing is applying image analysis techniques to the OCT scans from the UK Biobank to do detailed retinal sublayer quantification. And they published a range of articles looking at different correlations and using this data. Now, one of the most interesting, in my opinion, was work that they published in JAMA Neurology in 2018, which showed that very small changes in thinning of the retinal nerve fiber layer on OCT scans were associated with both current and future cognitive decline. And so you can imagine the excitement within the ophthalmic community as we begin to disentangle these findings. Now, where I come in then is working with Praveen and Paul and others. We have a separate study called the ALSI study that I think is going to be very complementary to the eye and vision component of UK Biobank. Now, this is a study where we've taken patients at Moorfields aged greater than 40 years, and all of the ophthalmic imaging done at Moorfields Eye Hospital over a 10-year period, 2008 to 2018. And what we've done is we've combined that with the NHS Digital Hospital Episode Statistic Database, the national database of hospital episodes. Now, we've described the cohort profile of that in BMJ Open just a couple of months ago. And essentially, I'm just going to give you some initial numbers from that study. So that includes 353,000 patients at Moorfields, about 1.3 million linked hospital episodes, and includes about 6.3 million retinal images from Moorfields. Now, of the imaging that we have, that consists of about 1.8 million color fundus photographs and about 1.4 million OCT volume scans. So huge amounts of imaging, routinely collected imaging data. And the nice thing is that when we link those images and the ophthalmic data at Moorfields with the hospital episode statistic database, we can find all incident cases of cardiovascular and neurodegenerative and other systemic disease among patients who previously attended Moorfields. And so, for example, we have about 10,000 incident cases of ischemic stroke. And by comparison, in UK Biobank, there's about 700 patients with retinal imaging who've gone on to develop a stroke. And similarly, we've got about 12,000 incident cases of heart attacks, with about 990 comparable patients in UK Biobank, and 13,000 incident cases of dementia. And in UK Biobank, we've got about 250 incident cases of dementia with retinal imaging, and a range of other cardiovascular and neurodegenerative disease. So in particular, for example, we have 65 patients who've gone on to develop Huntington's disease who've had retinal imaging done at Moorfields. Now, we think that this is a very exciting study. Now, there's lots of reasons for that. The large amount of data that I've just described, the fact that it comes from a very diverse population and very different socioeconomic groups than in UK Biobank. But of course, there's lots of limitations to the routinely collected data that I've just told you about also. And so what's really exciting for me is the idea that we can combine this outside study with the data from UK Biobank in some way and take advantage of the genetic data, the neuroimaging data, and the other advantages of UK Biobank, the longitudinal follow-up data that's been collected, and see how we can combine them to get the best of the both worlds with the routinely collected data from outside. So could we develop models on the outside data set and externally validate them on UK Biobank, or vice versa? Now, in terms of next steps, I just want to highlight that much of the work that I've been talking about has been led by a PhD student who works with me called Siegfried Wagner, really a rising star in this field. But the reason why I think that this is potentially exciting is that with these large numbers of scans and with the power of deep learning, we may be able to pick up some of the signs of neurodegenerative disease or indeed other systemic disease from routinely done ophthalmic tests, such as OCT. In fact, OCT imaging is now done routinely on the high street, and approximately 50% of all optometrists within the United Kingdom offer OCT scanning as a routine part of the examination. Now, we think that that's interesting because we know that the figures for NHS health checks suggest that only about 9% of people between the ages of 40 and 59 take up this health check. Whereas in a similar population, about 80% attend primary care during that time period. So just to say, we hope with this work to try and reinvent the eye examination for the 21st century. And then if anyone would like to get in touch with me, that's my email address. Thank you very much.
Lovely, thank you. Thank you very much. Well, there really are exciting developments there, and I'm looking forward to seeing more of them. I'd just like now to introduce Naomi Allen. We all know Naomi as the Chief Scientist for UK Biobank. She cut her epidemiology teeth, as it were, on cancer epidemiology. And I think she does an absolutely fantastic job in helping us as scientists engage with the complexity of UK Biobank data. So thank you for that, Naomi, but over to you for assessing change over time.
Thanks very much, John. So I'd just like to start by talking a little bit about what is the value of having repeat measures in a large study like UK Biobank. So in contrast with the germline genome that does not vary over your lifetime, exposures to lifestyle factors, environment, and endogenous biomarkers such as circulating levels of metabolites and proteins may vary quite considerably within an individual over time. And this is due to changes in exposure levels, various risk factors, the aging process, and other biological processes, particularly those related to early pathology. So taking a single measure at recruitment into UK Biobank may not accurately reflect long-term average exposure to a given risk factor, which may in some circumstances lead to a substantial attenuation of an exposure disease association. And so if we were to have longitudinal measures that capture change in exposure levels over a long-term period during an individual's midlife, these would be a far more powerful predictor of disease risk than if we were to rely on a single measure taken at baseline, particularly if it doesn't accurately measure long-term average measures. So what serial measures do we currently have in UK Biobank? So obviously at recruitment between 2006 and 2010, where for 500,000 participants, we collected a vast amount of data, as you all know, on lifestyle, physical measures, and biological sampling. And then in 2012 and 2013, we performed a repeat assessment on a subset of 20,000 individuals, specifically to address regression dilution bias. And this is caused by the measurement error that I've just spoken about when you use a single baseline exposure. And this error can be taken into account if you use repeat measures taken from a reasonably representative subset of the cohort. And this is exactly what this repeat assessment was designed to do. Then in 2014, we first started doing the pilot for the imaging assessment of up to 100,000 individuals by the end of 2024, which, as has been mentioned by Nick and Paul, includes MRI scans and other imaging related modalities, but it also included a full repeat of the baseline assessment with a few minor exceptions. And then in 2019, we then first started to bring people back for a second imaging assessment, initially in up to 10,000 individuals, and as Nick said, with plans to scale this up further over the coming years. And the purpose of this second imaging assessment is principally to enable researchers to assess changes in imaging-derived phenotypes, for example, to enable research into early disease markers such as structural changes in the brain that might be related to cognitive decline and subsequent dementia, or to enable researchers into looking at the determinants of physiological change, such as a change in bone density from the DEXA scan or loss of grey matter in the brain, for example. But as you can see, so we have up to possibly 4 serial measures in relatively small subsets of the cohort. And what would be really ideal was if we could perform a repeat assessment of the full baseline visit for as many of the surviving participants as possible. This would have really high scientific value because it would enable assessment of changes in risk factors, including lifestyle, biomarkers such as metabolomics, proteomics, but also methylation markers, assuming there was appetite to do long-read sequencing by nanopore, as Martin Tobin suggested this morning, on the baseline samples and these repeat assessment samples. And this would enable assessment of changes over about a 15-year period of time, which would be much stronger predictors of disease than relying on a single baseline measure taken at recruitment. So, for example, it would enable researchers to assess how changes in lifestyle and metabolomics and methylation are associated with mental health outcomes and dementia, for example. It would also enable assessment of changes in functional health outcomes. So, for example, researchers could look at what are the determinants of age-related cognitive decline in the cohort, what are the determinants of a reduction in lung function or hand grip strength. And again, these would be much more strongly associated with previous exposures than relying on a single assessment of these health outcomes taken at one specific point in time. So what's critical is, if we are going to do this cohort-wide repeat assessment, is to do it now while the cohort is still relatively young and mobile. I mean, the participants are now in their 50s to their 80s with an average age of about 70. And we think that of the 460,000 surviving participants in the study, we could potentially recruit up to about 300,000 of them, assuming that we could get participants along to an assessment centre that was relatively local to where they lived. Because what we know is that the greatest participation rates are among those participants who have to travel the least distance to any given assessment centre. So we know that about 20% of the cohort have moved since recruitment, but actually they haven't moved very far from their original residence. So we think it's entirely feasible that we could recruit up to 80 or 90% of the cohort could live within 10 or 20 miles of one of these temporary assessment centres. Of course, if we did do a repeat assessment on as much of the cohort as possible, it also would provide a really unique opportunity to do more targeted assessments of ageing and dementia. So, for example, we've been thinking, and these additional assessments, if we were to include them, would obviously need to be accurate, valid, easy, and quick to administer. So we've been thinking of whether we could include physical measures that were specifically related to strength, mobility, and functional fitness to enable researchers to assess frailty in a more accurate manner. So, for example, could we do a gait analysis that assesses walking speed and step length, a chair stand test that could measure leg strength and endurance, or a balance test, which would be important for assessing the likelihood of falls and fractures, all important for assessing frailty. Could we include a transportable DEXA scan that could travel around the different assessment centres to assess measures of changes in bone density, obviously important when assessing osteoporosis and the risk of osteoporotic fractures. And the DEXA scans that we've already seen also provide excellent measures of body and muscle composition to assess age-related sarcopenia, and also calcification of the abdominal aorta, as Nick was talking about earlier. Of course, if you've got participants coming along to an assessment centre, that also enables us to perhaps include additional cognitive function tests that can only be done face-to-face. So, for example, could we include tests to assess visuospatial perception, or that assess vocabulary by asking participants to describe a drawing, and then that audio recording is then extracted for various aspects of speech, such as the speed of the speech, the vocabulary, the rhythm of the speech, all of which are important aspects of cognitive decline. And of course, if we do a repeat assessment on everyone and bring them back into a centre, that does provide an opportunity for an additional biological sample collection. So is it worth us collecting skin cells or stool samples or additional fresh blood samples that may be needed for certain assays that are specific to ageing and dementia? So my final call is actually to please do get in touch with us if you have ideas about what we could include, whether that's giving people physical activity monitors when they leave the assessment to assess changes in sleep and physical activity, what additional biological samples could we include, other physical measures related to frailty. I mean, UK Biobank is your resource. We're just here to build it and to make it as relevant as possible to enable all different types of research to be performed. So I'm really excited, and I hope you inundate me with emails about different types of assessments we could include. So thank you very much, John.
Okay, well, thank you, Naomi, for that call to action. I hope as the scientific community we respond. Now, before going on to the panel, can I just remind people that we are inviting questions from the virtual floor. So if you'd like to use the chat function to send us your questions for this eminent panel, this is not quite a once in a lifetime opportunity, but it really is a big opportunity. So please take the most of it. Now, I'd just like to introduce Hal Barron. Now, Hal is our sort of discussant, so agent provocateur, and Hal has worked, with Roche, GSK, Calico, Verily, and is now with Atlas. So he's in a really strong position to advise us on where we might go from here in regard to aging. Hal, as you think and reflect on what you've heard, a particular issue of interest to UK Biobank is what we could add to the repeat measurement to enhance its value.
Yeah, thanks, John, and wonderful talks by everybody. What I thought in listening to all of the talks, and not in just this session, but earlier, I thought I'd try to summarize to some extent how I think about using the UK Biobank or observational data in general, and maybe with that highlight three themes I've heard in this panel, just for to summarize and provide my own perspective. I think the first, and maybe it was said by somebody, but not in our panel, but really to fully understand the pathophysiology of human disease, and I think to understand how to find new therapeutics with a higher probability of success. We have to, I think, realize the inconvenient truth that the human has to be the model organism. We've spent a lot of time trying to model human disease with animal models, and I think the UK Biobank has provided an opportunity to do just that, to use the human as the model organism. Because when you have these large observational data sets that are deeply phenotyped, particularly deeply phenotyped with genetic information, you've heard many talks today about how you can use that data to do GWAS, to use Mendelian randomization, to give a sense of what variant is associated with what phenotype, and to use that information to find new targets that might be involved in the pathophysiology of a disease. And we have explored this in depth. I know other groups have as well. And when targets are identified by using this approach, human genetics, GWAS associations, and Mendelian randomization, there's at least a doubling in the probability of success for these targets. And so at GSK, and I know at other companies, we're using these incredibly powerful data sets. I think UK Biobank, which is the best in the world, to really redefine how we understand the pathophysiology of disease, what's in the causal pathway in the human disease, and therefore how to find the best targets. And I think with the increasing omics, I'll just use that broadly, that are being employed in the UK Biobank and other observational data sets, these data sets are becoming richer and richer and more informative in terms of these targets. Now, my own perspective is that despite this, I think something around 80–85% of the GWAS targets that are identified, we really don't know the function. We know that a base pair change results in a phenotype. And what's amazing in the last decade, particularly the last 5 years, has been the power of CRISPR and other functional genomic tools to, at the in vitro level anyway, take human mammalian cells and interrogate the impact of a base pair change, to see if the base pair is causal, maybe it's in LD with something causal, and to understand what is the variant's function. That, of course, is important, not for predicting risk, but for in terms of understanding the pathophysiology. And more of this genetic, genomic, I hope that we'll see more epigenetic data collected to understand how the gene and its transcribed, translated, and ultimately the proteins that are generated impact disease. But we've heard a lot of talks about how useful this can be. And of course, clinical covariates are being added at an incredible scale, both digital clinical covariates as well as enrichment of interesting aspects of human disease. I think the next phase of the UK Biobank is going to be, and many of the topics covered today touched on this, that human disease is very heterogeneous. Clinicians, I'm a clinician by training, cardiologist, and we tend to be overly simplistic in categorizing diseases. They're very heterogeneous. And as we learn more about the pathophysiology of disease, we can start to deconstruct this heterogeneity. And that's particularly important because for drug development to be successful, not only do you have to have the right target, but you need to have that target modified for the right patients. And every patient's pathophysiology might be the same pathophysiology or might be very different. Most diseases have a heterogeneous component, and therefore modifying one pathway might not work for one patient, whereas it would work for another one. And I think the UK Biobank and these large data sets will allow us to deconstruct this heterogeneity in ways that have been described today. And that will give us not only better targets, but the right patients for those targets. The two features that were commented on during these 4 talks that were really wonderful, I'd like to highlight. First of all, imaging. of all the omic data or all of the covariates, I should say, that are being measured, I think there's no doubt that imaging offers some of the richest data. It's massively high dimensional, as Pearse points out, where there's semantic representation behind even pixels that humans are not even remotely able to see or understand. And with these advances in deep learning and machine learning techniques that can deconstruct these relationships, whether they be at the genetic or clinical covariates, but when there's massively high dimensional data embedded in data sets, particularly images, lots of things can be observed, like Pearse's argument about being able to identify gender or age or body mass. They seem trivial because you can measure them, but the point being really important, this is embedded in the pixels, and this is profoundly interesting, because what we'll start seeing is this heterogeneity of disease will be deconstructed in a very profound way. And this will allow us to see better targets and the right patients and progression. We talked a lot about the importance of longitudinal data as well. In addition to imaging and longitudinal is even more powerful, but just longitudinal data in general, as Naomi pointed out, really powerful because each patient's their own control. As Nick said, the more power. You can imagine that somebody might present with a fasting glucose of 110 when they're 50, but every 5 years it's still 110. If you measure that over 30 years and they're stable at 110, they may have a reset problem, and they're not going to develop diabetes. Another person could be 90 when you first measured, but they're going up 5 points per year. Those people are progressing at a much faster rate. And understanding that progression is going to be very important, because the static nature of a one-time image or one-time assessment of any covariate loses enormous richness if you can't follow them over time. So I think imaging, all the omics in general, but imaging in particular, times longitudinal, is going to be very synergistic in understanding both targets and heterogeneity of disease. I'd like in just a few minutes just to point out one thing that I think Naomi was touching on. I'm going to complicate her point a little bit, because I think it's the next version of where we take this medicine and biology, which clearly are becoming a data science. Where is this going next? And I think it's in prevention. And in order to prevent disease, we have to know which patients are going to develop which disease. And I think these polygenic risk scores and other covariates that can be added to give a group a risk are very useful, and they're a very good beginning. But at the individual level, they don't really help. You have a 6% risk of having heart disease if your Framingham scores, blah, 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 and you can optimize these with genetics. But at the individual level, you're still on a very uncertain basis. And even with Mendelian disorders, there's incomplete penetrance to the point where even, I don't know, like a GBA1 mutant still only has a 15% or so risk of developing Parkinson's. I'd like to throw out something that Naomi alluded to, which is, how do we stress the system? My view of clinical disease is that it's the failure to maintain homeostasis. When you're young and your cells are robust and your organs are robust, you can withstand insults because you are able to maintain homeostasis at, like I said, the cellular or organismal level. As you get older and more stresses emerge and more and less fidelity of the system exists, your buffer capacity, as I like to refer to it, diminishes. And if we were able to measure a person's buffer capacity for a given disease, we might be able to identify another set of covariates that is as rich as imaging and as rich as longitudinal. And to some extent, longitudinal data is really saying, what if we stress the patient with time? That's really, in my mind, what longitudinal data is. What's the impact of time as a stressor? But unfortunately, that stressor takes time. What if we could do it when they're sitting there? I'm a cardiologist, as I mentioned, by training, and one of the things that we routinely do in clinical cardiology is stress the patient. Someone could present with chest pain and a normal EKG, and we find that to be useless, because what you need to do is see them respond to exercise on a treadmill to see if their ST segment's depressed, their vascular reserve. You can see this with glucose tolerance testing to see if someone can withstand a challenge of glucose. Naomi mentioned a 6-minute walk or hand grip. This is a form of stress. You're asking people to perform a test. I would love to know what Pearse might think would be a great stress test for the eye. One could imagine a bright light and seeing how long it takes for that image to remove. You could, I'm sure, not an ophthalmologist, I don't know. There's plenty of stress tests. I would love to hear Paul's thoughts about stress testing the brain in terms of cognition. How can we identify the reserve? And this is different, I think, than something Pearse was mentioning, is that there might be preclinical disease. Preclinical disease is where you can identify a form fruit of the disease that's not yet clinically manifest. I'm actually talking about people who have no preclinical disease, but whose buffer capacity is limited, such that an intervention might actually prevent disease. I think that's something that if we can incorporate into the UK Biobank, very, very hard, very challenging, but my bet is over the next decade, there'll be more and more ways of stressing the system and actually assessing their reserve, if you will, their buffer capacity, their ability to maintain homeostasis, to identify who's really at risk for what disease, so that when we get these treatments, we can actually stop just treating to modify disease, which is terrific and wonderful. And I've spent 25 years doing that, but I would love to see the next 25 years of medical science focus on preventing disease. And to do that, we have to have a much better sense of who's at risk for what thing, and maybe the targets for reserve. This is the last point I'll make, and I'll stop. Maybe the pathophysiology of how you increase buffer capacity, how you actually regain homeostasis, they may be different than the pathophysiology of the disease they've caused. So in other words, there may be ways of improving lysosomal health, which might end up being a way of adding buffer capacity to the brain. That's maybe not necessarily what's causing Alzheimer's or something like that, but this may open up new targets again for prevention. So wonderful resource. We've clearly entered a phase where medicine and biology as a data science, large, deeply phenotyped observational studies, the UK Biobank being the premier, is a resource that is incredibly invaluable for so many things, for new targets, for annotating the targets to understand their function, imaging and longitudinal data being the most important next advances. And I'll throw out, as I said, the phase after that, which might not be for another decade, is stress testing, reserve, and maybe with that, just stop and see if we have questions from the audience or maybe some comments from the panel.
Well, thank you very much, Hal. I think that was very provocative. I'd just like to ask the panel what they think of stress testing the brain.
Well, John, I'll answer that. I mean, it's a tremendous idea, and it is one that is part of some batteries of tests, particularly divided attention tests. There also are tests associated with accelerated forgetting that tap into this, and it has been done in the context of various electroencephalographic tests. I think this is exactly right. It's a way of inducing a phenotype when it's not present in a baseline state, and we can begin to see it. I think in relatively easy ways. None of these tests take very long. So I think enhancing this along with addition of some of the digital tools in the biobank follow-ups, all of these things can be done offline and can leave time for other sorts of measurements with the in-patient or in-person visits to the participant centers.
Any other thoughts?
It just made me think, Hal, when you were talking about stresses, that at recruitment, at least for the last 100,000 participants or so, we had them perform a cardiorespiratory test on an exercise bike that was graded exercise, and we measured their heart rate, and that's a really good example of a stressor. But I think in the back of my mind, I was thinking, well, these participants, their average age of 70 now, we don't want to stress them too much. We don't want to feel that they run a marathon. But I think it's an excellent idea in terms of what tests can we include that are feasible, acceptable to the participants, but where we still get a really good handle on what are their reserves and how quickly does it take for them to get back to homeostasis. So it's a really neat idea.
Yeah, and maybe one comment for Pearse. I've seen some data on being able to differentiate various shades of gray, and how that predicts longevity and even cognitive abilities that might be reflective of, it's not really a stress test in the sense that it's stressing the system, but it's looking for your ability to differentiate things that are stressful to see the difference between. So, I mean, to your point, I don't think all stress tests have to be, increasing risk in any way. There's ways of asking questions or things that are part of normal life that you can ascertain their ability to differentiate.
Nick, could I ask you what you think might be the difference between a cognitive test, which is a stress test, and a cognitive test?
Yeah, so you have to be careful, I think, what we're trying to achieve here. I'm not sure about the cognitive test, but let me just illustrate this in terms of kidney function. Okay, so kidney function deteriorates from a very young age, and it's not until you get very severe loss of kidney function, I mean, 90% or more of normal kidney function, you start seeing manifestations of that. And so it's not really the ability to stress your kidneys, because I think life stresses your kidneys, and having a test, what that test would do is just make the pathophysiology associated with slow decline in kidney failure more present. Okay, so I think what the point we were trying to make is that if you have a very sensitive measure of the consequential pathophysiology, then life itself is the stress for that system. Does that make sense? The simple example there would be glucose tolerance. Okay, we do glucose tolerance because we have to stress the system in order to make the measurement. But if you have very sensitive ways of measuring instant secretion, for example, you don't need to stress the system with age because that inherent deterioration is detectable. Again, I'm not saying I think the idea of buffering to get back to homeostasis is exactly what we should be thinking about. I'm not totally convinced that you need stress tests in order to do that. I'm more convinced that you need sensitive readouts in order to do that.
Yeah, I think it's a really good point. And I'll just add that when you understand the pathophysiology of disease perfectly, which we never do, but the closer you get to that, I agree with you. When you think about heart failure as an example, and particularly HFpEF, diastolic dysfunction, there are many people by echo who have exactly the same disease, some of whom are symptomatic and not, even though their LV thickness and their relaxation properties seem identical. If you would infuse them with saline or blood or some stress test, you would be able to differentiate those two despite all available technologies being able to not see the difference. Most diseases, we don't know it as well as kidney function, where you're right, there isn't buffered, there's no way to buffer your kidney function. Your creatinine goes up, functions down, there's not a lot of buffering from other organs. But for many organs, there's a complex interrelationship between the immune system or the vascular system or so many things that the individual measurement of that organ might not reflect the other organ's ability to, like the adipocyte is to the pancreas or the brain is to the gut. And because of this complexity, sometimes stressing the system might actually be able to see this massively high dimensional complexity unraveled, which is.
But part of this too is that this is a specific way of looking at dynamic data, and I think one of the really great opportunities for UK Biobank going forward is to collect more serial, increasingly fine-grained dynamic data, either using digital tools, serial follow-up, multiple blood markers even within a day. All of these can be hugely informative, as has been shown from smaller individual studies.
From the point of view of disease heterogeneity and repeat measurement, to what extent could UK Biobank become, if you like, a dementia progression cohort, a disease progression cohort, going from the very earliest genetic risk through to diagnosis and death?
Well, John, at the risk of just saying something again, I mean, I think it's well positioned to become one of the world's premier studies in this, recognizing its limitations in terms of populations. And that's simply because there are few studies that have followed people for such a long time in such depth and will continue to follow people towards the latest stages of cognition. To pick up on Hal's point, I think this is really one of the brightest futures for dementia therapeutics, and that is in trying to understand what the factors are that can influence differences in the trajectory of cognitive impairment to prevent the onset. An often quoted observation from the epidemiological literature is that even delaying the onset of dementia in the population by 5 years a rather modest goal, nothing to do with cure, would have a huge impact on the prevalence of the disease in our population.
I think it's just also worth adding here that with the repeated measures of cognition from our online questionnaires, in addition to the cognitive function tests that were done at the assessment centres, for some individuals, you've got really quite granular and repeated measures of various aspects of cognition over a 10-15 year period. And so I think already the cohort is very well placed to be able to assess age-related change in cognition and change in cognitive impairment over time, even now, even if we don't do repeat assessments, in the future. So I think, using the UK Biobank resource now with all the data that it has, it's certainly well placed to look at cognitive impairment over time.
Nick, any further thoughts on that briefly, and then perhaps we could ask Pearse?
Yeah, Pearse, take it away.
Pearse, over to you.
I'm not sure that I have anything immediately intelligent to add to what Paul has just said.
Okay. I mean, one of the issues is obviously getting disease-relevant biomarkers, blood biomarkers, and I think there is a whole debate to be had about what would be appropriate, and there's plenty of blood available for that, so I'll leave that. Folks, can I just thank you very much for a really interesting and stimulating debate. Thank you, panel, and thank you all the presenters.
Thank you.
Thank you.
Well, I want to thank all the speakers in this session and the previous sessions, but also particularly the people who've joined us for this conference. It was very disappointing for us that we couldn't have an in-person meeting in London this time round because of the national rail strike here in the UK. On the other hand, some 2,500 people from around the world have joined this virtual meeting, and it was fascinating to see where they were, from Australia, Japan, China, Indonesia, Philippines, Malaysia in the east, through to Saudi Arabia and India, then into Europe, obviously the UK, but Sweden, Italy, Germany, Switzerland, Hungary, Turkey, Ireland. Then into Africa, we had Dan Stein talking from South Africa, but also researchers from South Africa, Kenya, Uganda, Nigeria, and from the Americas, the US, Canada, Chile, and beyond. So although 500 of us couldn't be in London together, 2,500 of us have been together through the day. The message I really wanted to get across was that the job of the UK Biobank team is to build the resource, to develop the resource in the ways that make it of most value to the widest possible research community for the widest possible health-related research. And if we can convince you that this is your resource, that it's not just something for you to use as a research community, but really the success of UK Biobank depends, yes, on the imagination of the ways in which the resource is used by researchers around the world, but also in your imagination in terms of what else should we be doing to enhance it. And there has been an enormous amount of input from many different groups in terms of how to enhance the resource. You've heard from many of them today, from genetics to imaging to remote monitoring. But what we want to get out of this meeting ourselves is what enhancements of the cohort would add further value. You've seen the value of extension of assays from genetics into proteomics, and perhaps now into other omic assays. Well, what assays? Some of these have been funded by industry, but also by charity, by government. And And some of them being driven not just from the commercial sector, but from academic researchers. So the telomeres, which were funded by the Medical Research Council, done in Leicester by Nilesh Samani and his team, and a group in London that have got funding for infectious markers from philanthropic support. We're looking for ideas for turning these samples into data. The completion of the initial imaging and repeat imaging, and it was really the driving force of the eye consortium that introduced optical coherence tomography and eye assessment at the baseline, that have also pushed for that to be in repeat imaging and have helped to support that project. But there are other ongoing discussions around other sample collection that might occur at that visit. And we're hoping that that will occur, that will allow us to have assays beyond the kind of the omic assays, particularly into single cell sequencing. At the moment, the linkages into the health outcomes are limited into death, cancer, hospitalization, primary care for COVID research, and we hope that will be extended. But as you saw from Naomi Allen's talk, the opportunities to link into many other kinds of health record data are really huge. So again, we can link into them, but we need expertise to turn those data into validated specific health outcomes. And I think that's where the informatics expertise in AI and machine learning will be particularly interesting. And I think that the possibilities of doing challenges to help that. Remote monitoring, again, Aidan Doherty talked about that. We've used UK Biobank specific monitors, like sending out accelerometers, but would it be possible to access commercial monitors that people already have? Would it be possible to add phone apps that we can get continuous monitoring of participants? And can we use existing data sets, such as web search data, to get additional information about participants? Finally, the discussions we just had about repeated assessment of as many of the surviving participants. What should be done at those visits? You heard some of the discussion, but I'm sure there are many more ideas out there as to how one could assess changes in cognition, frailty, aging, etc. Should there be imaging of some kind at those visits that could be done at scale? And then finally, the thing that Naomi touched on about extending to first-degree relatives. We found in the COVID period that going out to participants and asking if they could get their children or grandchildren to provide samples was very successful. Could we do that on a massive scale and get at least genetic information from first-degree relatives to enhance the cohort? But what else? And that's really the challenge to you. What else should we be doing and how should we go about doing it? And then finally, the topic we touched on with Dan Stein's presentation was how best to encourage wider use by a more diverse population of researchers. He was talking about extending to other parts of the globe, and we're very keen to understand how best to do that. But also, how do we extend to other parts of the research community beyond the biological researchers into experts in bioinformatics, social science, environmental science, all of those other areas? How do we introduce UK Biobank to them so that they can help to draw more information about health and the ways to prevent and treat disease? Again, how else should we be extending the research community? So I do want to thank everybody for your contributions to UK Biobank in helping to enhance it, in using it, in generating really interesting research out of it, but also inviting you to tell us what more could we do. Thank you very much, and I'll now pass over to Rob Buckle, who will close the meeting.
Thank you very much. It's a privilege to be invited to close today's meeting on behalf of the Medical Research Council. By way of introduction, I'm Rob Buckle, MRC's Chief Science Officer. And as you'll be aware already, Biobank is a flagship UK investment which we co-fund with Wellcome, the Department of Health and Social Care, British Heart Foundation and Cancer Research UK. And I'm sure I speak on behalf of all the funders when I say that it's been a pleasure to see what UK Biobank has achieved and showcased so successfully today. Biobank holds a unique position globally, and I think the figures of engaging 27,000 researchers across 90 countries speak for themselves. And UK Biobank is undoubtedly a beacon for open science and a pioneer in so many new ways of working. Due credit to the Biobank team, and Rory in particular, for turning the original vision into a reality that's far exceeded the original expectations of its founders. So if I can briefly start with a brief comment about MRC and our positioning in relation to population health research, and then I'll move on to offering a reflection on UK Biobank. Most of you from the UK will already know that MRC is part of UK Research and Innovation, and that UKRI is a non-departmental public body sponsored by the UK government. We bring together 7 research councils, Research England and Innovate UK, and work together to deliver an ambitious research agenda, drawing on a depth and breadth of expertise and diversity in the portfolio. Now, one of the strategic aims of the newly published UKRI strategy is about securing better health, ageing and well-being, including research into prevention and population health, and in order to identify sustainable ways of preventing non-communicable diseases alongside interventions that will improve population health and reduce health inequalities in the UK and beyond. MRC itself will be publishing its own delivery plans soon, and again, population health, ageing and well-being are central to those plans. So we feel we're well positioned within UKRI to take a more holistic approach to population health. And it's particularly good to see, for example, from today, the developments on environmental exposures that we heard about within UK Biobank, and the stretch that's going beyond the biomedical to address wider determinants of public health. And looking more broadly at the UK funding landscape, the potential of Biobank is well recognised in the UK government's strategy in terms of health and research, for example, the Genome UK strategy from 2020, the 2021 Life Sciences Vision, and the NHS long-term plan. So where do we find UK Biobank today? Well, MRC and Wellcome are the long-standing partners who have supported UK Biobank since its inception in 2006, along with the other funders. Its creation was seen as high risk, perhaps strangely from where we stand now, and the decision to establish Biobank was not without controversy, given the lead time before outputs could be expected and the ethical hurdles that needed to be surmounted. But it's clear that any doubts at that time were misplaced. UK Biobank has grown into a truly world-leading and unique research resource. And indeed, we're now seeing exponential growth in the requests for data use, both nationally and internationally, as well as the numbers of publications, patents, and policy outputs, and coupled, quite frankly, to quite unanticipated levels of commercial interest in this resource. But the sense of achievement goes beyond the metrics. In particular, it should be noted how Biobank has led the way in driving 21st century approaches to epidemiology through the integration of molecular and big data approaches to population health, and through embracing new cutting-edge technologies for data collection and analytics, and not least through the adaptation to changes in participant and societal expectations over the course of that time. This has been achieved through the commitment, dedication and professionalism of a gifted team, and as its figurehead, Rory has been widely recognised for his visionary leadership and drive, which has been provided over the course of the past 20 years. And indeed, in 2020, Rory was awarded the prestigious MRC Millennium Medal for his transformative national and international contributions to both cardiovascular disease research and, of course, UK Biobank. So Biobank's been remarkably successful in creating project-specific industry partnerships, and these have enriched the resource by providing an unparalleled, rich and deep genomic and phenotypic data at scale. The whole genome sequencing work in particular is a major success story, and this year we are set to see the UK Biobank secondary imaging programme recommence following its interruption due to the pandemic, which it will overall finish in 2029. We've reflected today on how that repeat imaging is pivotal to understanding how early changes in the brain and the body are associated with the onset of age-related neurodegenerative disease. We've also heard how plans have evolved and that OCT imaging, for example, may get added to the list of modalities in play. The imaging work as a whole, together with the sequencing and the scale that this collective activity embraces, as well as the access to the surrounding lifestyle context of participants, puts UK Biobank in a fantastic position to allow researchers to understand the origins of not only dementia, but other diseases of aging. And we are all looking forward to hearing more of this activity as it advances. Similarly, the multi-omics story is just beginning, and will get us still closer to understanding gene function at the physiological level and how such biological systems can unravel in disease. And this has the potential for wide-reaching impact for the future development and testing of new therapeutic approaches. UK Biobank sets high standards and aims to be a front runner in open access, such that researchers can interrogate the breadth of the resources-rich data. As you know, Biobank doesn't do research itself, it enables research for everyone else. And in doing so, it's provided amazing opportunities for scientific innovation and research into the determinants of health, well-being and disease. And in this context, I was particularly pleased to see the efforts that are being undertaken to support access for early career researchers and also researchers for lower middle income countries. I was also pleased to see the session on deepening participant involvement and look forward to seeing the story unfold. Without the tremendous and sustained commitment of Biobank's participants, the research could not advance in the new and exciting ways that is enabled through this resource. I'm sure that Biobank and the participants will increasingly engage in purposeful and meaningful ways so that the participants' wisdom can enrich approaches taken by the resource. And another noticeable feature that we heard today is the agility with which during lockdown, the team moved to use the resource to support COVID-19 research, including research through linkage with primary care records. As well as being impactful in helping address the pandemic head-on, this has also shone a light on ways to address the broader barriers that exist within the health research ecosystem, and which I hope can be captured and learned from. So finally, what of the future? Well, the release of the first tranche of whole genome sequencing data last year and the further release next year will undoubtedly transform the way in which researchers study genetic determinants of a wide range of health outcomes, which itself will fuel numerous other projects over the coming decade. And given UK Biobank's growth and successes over the last 20 years, it is very well placed now to contribute even more to the wider research community. For example, through drawing in wider disciplines, such as the socio-economic or environmental perspectives that we've heard about today. And of course, working in partnership and adopting technological innovations, which has already been instrumental in the development of a number of UK Biobank projects, such as the whole genome sequencing. Beyond the scientific opportunities, the capability of Biobank to respond to increasing interest and demand will be enhanced through its move to central Manchester, where the potential links across the science park and the wider university should herald a new era and more sustainable phase for Biobank's operations. This will be enabled by £128 million of UKRI infrastructure funding over the next 10 years, as was formally announced last week. And as part of its legacy, the UK Biobank way of working and learnings will critically inform the establishment of new UK activities in population health research, where, for example, MRC and ESRC are initiating new national cohort studies in both early life and adolescent life, as well as an open science resource, Population Research UK, which will ensure we can fully unlock the potential available through linking population and administrative data sets across a multitude of studies. So let me finish here with saying what a great day this has been, and to thank Rory and the wider Biobank team for taking us on such an incredible scientific journey, and with the promise of so much more to come. Thank you also to everybody for participating today, and best of luck with your future research plans. And with that, I'll wish you all goodbye. Thanks very much.
So the thing that I always want to say to people who take part in any study, whether it's Biobank, whether it's a clinical study, is just a big thank you, because it's a very generous act. It's one where people understand that you can be part of creating knowledge that will benefit other people.
Without people donating their samples and participating in clinical research or research, we don't progress in terms of our disease understanding, and our medicines will not be as effective. And without you, our breakthroughs will take longer, and innovation will take longer.
Why am I a participant? For the same reason that I wanted to be part of driving UK Biobank. I think that it is a fantastic opportunity to change the way we prevent and treat disease, and I wanted to be part of that in every way I could be.
My deepest gratitude and thanks.
Thank you.
Thank you. So it's huge thank you to those volunteers. They are fantastic.